Good morning, students. This session for second BSc Biotechnology Semester 4. Title of the paper is Plant Biotechnology. Today we are going to discuss the topic is synthetic seeds or artificial seeds. Here the synthetic seeds are nothing but it is a encapsulated method, nothing but immobilization of enzyme. Here, the plantlet conversion from encapsulated somatic embryo. Elongated torpedo stage somatic embryo is completely it is arrested by a enzymatic membrane, nothing but it is a chemical encapsulated by using sodium alginate with calcium chloride. Then they have producing a shoot and root. Then it was dipped into the soil. Artificial seed can be defined as artificial encapsulation of somatic embryos, shoot, bud, or aggregates of cell of any tissues, which has the ability to form a plant in vitro or ex vivo condition. Artificial seed have also been often referred to as synthetic seed. So artificial seeds can be defined. It is artificial encapsulation of somatic embryos. In this artificial encapsulation done by two chemicals like sodium alginate with calcium chloride. It is otherwise called as synthetic seed, artificial seeds or synthetic seed. Going to the history, artificial seeds were first introduced in 1970s as a novel analog to the plant seeds. Before 1970s, nobody can introduced this type of seeds, plant seeds. The production of artificial seeds is useful for plants which do not produce viable seeds. It represents a method to propagate these plants. So, here these artificial seeds are used in the multi multiple copies. That means same character in micropropagation methods. In micropropagation method, in a single crop, we produce number of clones. Here, it represents a method to propagate these plants. This propagation is nothing but micropropagation. Artificial seeds are small sized and these provides further advantage in storage, handling and shipping. These are very small sized, very small sized. It look like as a artificial artificial water balls like shape. So these are the term ambling is used for the plants originated from synthetic seeds. Here, the use of synthetic varieties for commercial cultivation was first suggested in maize using the scientist is 1919 Hayes and Garber. సో మేజ్లో దీన్ని ఫస్ట్ కమర్షియల్గా ఇంట్రడ్యూస్ చేశారండి. దెన్ ద ప్రొడక్షన్ ఆఫ్ లేదా ఆర్ ప్రొసీజర్ ఫర్ సింథటిక్ సీడ్ ప్రొడక్షన్. ఫస్ట్ ఆఫ్ ఆల్ వీ హ్యావ్ ఎ పర్టిక్యులర్ culture media. On that culture media we can grow the explant from a somatic embryo. On that somatic embryos are isolated, they are mixed with sodium alginate solution in a beaker. Then we are encapsulated embryos with calcium chloride, that is 30 to 100 millimolar calcium chloride solution or calcium nitrate solution. Then here when we are adding the calcium nitrate solution upon sodium alginate mixer, each and every drop to produce a bubble shape. So they are called embryoids. These embryoids are stored in a greenhouse. These embryos are stored in a greenhouse. Then beads are vermiculate, then germination after planting the pots. So going to the method for production of artificial seeds. Here establishment of callus culture. The callus culture establishment, then it will induction of somatic embryos in callus culture, then maturation of somatic embryos, synchronization of singulation of somatic embryos, mass production of somatic embryos, standardization and encapsulation of somatic embryos, mass production of synthetic seeds, testing embryos to plant conversion, and then they are planted upon greenhouse or field planting. In this encapsulation of the somatic embryos, water soluble hydrogels are used for making artificial seeds. So, mainly useful the gels are sodium alginate with calcium salts, sodium alginate with gelatin, calcium chloride, kerosene, it is complexing agent like potassium or ammonia chloride. Then gel-right, it having the temperature fluid. Then two methods used for encapsulation method. The first one is the complexation via a dropping procedure. Isolated somatic embryos are mixed with 0.5 to 5% weight by volume sodium alginate and dropped into 30 to 100 micrometers in calcium nitrate solution. Surface complexion begins immediately and the drops are gelled completely within 30 minutes. So here first gel complexation via dropping procedure is sodium alginate mixed with the calcium nitrate. Here sodium alginate, calcium nitrate, both having by using the encapsulation method. Here every droplet can produce a seed. It produces a bead shape. It produces a bead shape. Second one is the molding. In this molding, isolated somatic embryos are mixed in temperature depending dependent gel such as gelrite and placed in the well of microtiter plate and it forms gel when the temperature is cooled down. So it is gel titer method. So it having gelrite. Then need for artificial seeds. Artificial seeds make a promising technique for propagation of plants, non-seed producing plants, polyploids with elytrials, and plants line with problems in seed propagation. Being clonal in nature, the techniques cut short laboratories selection procedure for conventional recombination breeding. Why we are using alginate? Alginate is hydrogels are frequently selected as a matrix of synthetic seeds because of its moderate viscosity and low toxicity of somatic embryos and quick gelatin and low cost. Agar is considered inferior to alginate with respect to long-term storage. Alginate chosen because it enhances capsule formation and protects somatic embryos against mechanical injury. So అగార్ అనేది కూడా మనకి జెలాటిన్గా కనిపిస్తుంది లో కాస్ట్. బట్ అగార్కి లాంగ్ టర్మ్ స్టోరేజ్ ఉండదు. ఇక్కడ ఆల్జినేట్కి మాత్రం లాంగ్ టర్మ్ స్టోరేజ్ ఉంటుంది. మెకానికల్ ఇంజ్యూరీని కూడా ఇది ప్రొటెక్ట్ చేస్తుంది. అగార్లో ఆ ఫ్యూచర్ అనేది ఉండదండి. దెన్ పొటెన్షియల్ యూజ్ ఆఫ్ ఆర్టిఫిషియల్ సీడ్స్. Here reduced cost of transplanting. Carrier of adjuvants such as microorganism, plant growth regulators, pesticides, fungicides, nutrients and antibiotics. It is used in large scale monocultures. That means a single type of plant can cultivated by more number, more number. It can produce the clones. Can be conceivably handled as seed using conventional planting equipment. So here some tools, analytical tools are recorded. They are production of large number of identical embryos, determination of role of endosperm in embryo development and germination, study of somoclonal variations. They are the potential use. The scope of synthetic seeds are: first one is the propagation, second one is the conservation, third one is the transport. In propagation, rare and endangered plants, allied genotype, genetically engineered plants, seedless plants, commercially important plants. It is used in the micropropagation methods. Conservation by long and short term storage in the germplasm. Transport, exchange of organic plant material free of undesirable contaminants. Advantage: high volume, large scale propagation method, maintains genetic uniformity of plants, direct delivery of propagules to the field, thus eliminating transplants, lower cost per plantlet, rapid multiplication of plants. The synthetic seed technology offers tremendous potential in micropropagation and germplasm conservation. However, further research is needed to perfect the technology so that it can be used on a commercial scale. Here, the limitations are limited production of viable micropropagation, asynchronous development of somatic embryos, lack of dormancy and stress tolerance in somatic embryos, improper maturation of somatic embryos that makes them inefficient for germination and conservation into normal plants. So by using this type of encapsulation method, we can produce the artificial seeds or synthetic seeds. They are widely used in the propagation methods for long storage by this type of completely encapsulated shell. That means shell is made. Yeah, on that on that shell, the cells are very live condition in their either shoot and roots are alive in conditions. Then when we are going to planting readily, these type of encapsulated seeds are planted by using the greenhouse or a soil. So it is widely used. So synthetic seeds are the artificial. It is otherwise called as artificial seeds. So artificial seeds have very demand today nowadays. Artificial seeds. So every droplet they mixed with sodium alginate and embryoids. It is drop by drop by using it is drop by drop in the calcium chloride solution. When the droplet comes into the calcium chloride, it becomes encapsulated bead formation. On that beads are nothing but synthetic seeds. They are called artificial seeds. Thank you students. మీకు ఏమైనా డౌట్స్ ఉంటే కనుక నన్ను కాంటాక్ట్ చేయొచ్చు. మీకు సంబంధించిన నోట్స్ కూడా మీ వాట్సాప్ గ్రూప్లో అప్లోడ్ చేస్తున్నాం. నోట్స్ కంపల్సరీ రాయండి స్టూడెంట్స్. థ్యాంక్ యూ స్టూడెంట్స్.